And what we're going to focus on in this lecture is population genetics, because this seems to be the trickier part of this chapter. Okay, so I have on here the definition of a population, and I had just gone over that, but the abbreviation SP is for abbreviation for species, and SPP is the abbreviation for species plural. So you may see that at some point. What we're going to be looking at when we look at population genetics is not the genes of an individual, but the genes in a population. And all of the genes in a population we refer to as a gene pool. And then we'll be looking at the frequency of different alleles within that population. So we'll be looking at, let me see if I can figure out where to get my pen. Here we go. Okay, so we're going to be looking at allele frequency, and that is the frequency of the different variations of the gene in a population. So here's a picture of that. And so within this population, we have 3 versions of gene X. So they're numbered gene version 1, version 2, and version 3. Now, in a diploid organism, or an organism that has paired chromosomes, one they receive from mom, one they receive from dad, they will only have 2 of those possible variations. So they can have 2 number 1s, they can have 2 number 2s, they can have 2 number 3s, or they can have a combination of 1 and 2, 1 and 3, and so on. So if we want to know the allele frequency, we would count up how many number 1s there are in this total gene pool, divided by the total number in the gene pool. And we could do the same thing for the version 2 and or the version 3. So that's what we're going to be looking at in population genetics, is the frequency of different alleles within a gene pool. Okay, so to work with these problems, we're going to use an equation that was developed by 2 scientists. So we have the Hardy-Weinberg theorem. And this is only really can be applied to a population that is not evolving, or where the gene's frequency is not changing. So in order for a population to have no change in their gene frequency, they must adhere to these 5 requirements. And so here's my little highlighter. Okay, so first of all, you have to have a very large population. If you have a small population, you can end up with a sampling error. And that will shift the genes if that sample does not represent the whole larger population. There can be no migration, either in or out. No immigration, no emigration. So we cannot have individuals adding from to the gene pool or removing themselves from the gene pool. Okay, no mutations. So there cannot be any random changes in the genetic makeup of these organisms. And the next 2 are kind of related, but we have to have random mating. So no sexual selection of organisms, so there can't be specific traits that females or males particularly find more attractive and therefore mate with specific individuals. And there can be no natural selection, so there can't be any adaptations that make, or variations within a population that make the one part of the group more fit in that environment than others. So if we were to have all of those held constant, we could have a non-evolving population. And the equations that we're going to be using is using these letters P's and Q's. So P is going to represent the, where's my, oops, I didn't mean for that to come in yet, but that's okay, it's here. Okay, so P is going to represent the frequency of the dominant allele. And Q will represent the frequency of the recessive allele. So we have P plus Q equals 1. So if we add the frequency of the dominant plus the frequency of the recessive, that should equal 1. Okay, so as an example here, we are going to, we need to find my pen. Okay. We're going to just say that we have 2 forms of a particular gene, and we're going to say big A is the dominant allele, and little a is the recessive allele. So if we're looking at a population instead of an individual, in that population, individuals will have either big A, little a, little a, little a, and so on. So big A and little a exist. So in the egg pool, there could be big A's, or they could be little a's. And so we're going to set this up just like a genetic cross that you've seen before. But in this case, we're looking at gene pools. So now the sperm can have the same alleles in their gene pool. And so if a sperm that's big A and an egg that's big A come together, we end up with big A. if a egg comes with the big A and the sperm with a little a, we end up with this combination. And if the if the sperm has a big A and the egg has a little a, we have this combination, and we have this combination. So this is the same as the Mendelian genetics that you did when you were in your first year of biology class. But what we're going to do here is we're going to add the frequencies of these different alleles. So we're going to pretend, in this case, that big A has a frequency, or 90% of the alleles in the population are big A. So we're going to give it a frequency of 0.9. And we're going to say then, therefore, little a has to have a frequency of 10%, because they need to add up to 100%, or frequency of 0.1. So 0.9, put it into my equation, plus 0.1 equals 1. So now when I set up my cross, I am going to include the frequencies of the alleles. So in this case, I have big A, and the frequency of that is 0.9. And little a, and the frequency of that is 0.1. Oops, like 1. Okay, and again, in the egg population, the frequency is the same. We're not separating out males and females here, the frequencies of the whole gene pool, and so therefore it's going to be 0.9 and 0.1. And now when I do my cross, and when I figure out what my individuals would look like, so I have big A, and now we're going to follow the laws of probability, which are on your equation sheet. And so the probability of an individual receiving both big A and big A is the product of the probability of each individual event occurring. So the probability of getting a big A is 0.9. The probability of getting a big A and a big A simultaneously is going to be 0.9 times 0.9, or 0.81. Like that. Okay. So here the probability of getting a big A from the mother and a little a from the father is going to be 0.9 times 0.1, or 0.09. Okay, and again, if the sperm is the big A and the egg is the little a, we would end up with, again, 0.09. And then finally, the probability of little a, we again take the product, so that would be 0.01. Okay, so the probability of an individual in this population being a homozygous dominant individual, if the allele frequency is 0.9 and 0.1 respectively, is going to be 81%. So 81% of this population would be big A, big A. Now, big A, little a, we're going to have to use another rule of probability, and that's when an event can happen in multiple ways. So we can have a big A, little a, if mom donates the big A and dad donates the little a, but we can get a big A, little a in another way, if dad donates the big A and mom donates the little a. So here what we do is we take the sum of these two. So the probability of this happening, the probability of that happening, so we take the two of those together, so that's going to be 18% of the individuals would be big A, little a. And then finally our little a population would be 1%. And now all of that, 1%, all of that should add up to 100%, and it does. So if we're going to replace this 0.9 here and say that that's a P, and so over here's a P, we're multiplying these two together, so essentially this is P squared, right? P squared. Over here, we're taking the A and the big A and the little a, and so this is the P, and this one over here is the Q. So here is really PQ, Q times Q, right? The frequency of the dominant times the frequency of the recessive. And also PQ here. And then over here, we're taking Q times Q, so this is Q squared. And now we have two ways of getting this, so this is going to be the big A. Little a is actually going to be equal to 2PQ. Okay, so things you have to memorize with this, or unless they're, I think they're on your equation sheet, you have to understand what they mean then. We need to know that P squared is the frequency of the homozygous dominant individuals. We need to know that 2PQ is the frequency of the heterozygous individuals. Q squared is the frequency of the homozygous recessives. We also need to know that P is the frequency of the dominant allele, not individuals. This would be referring to just one, not a diploid organism, just one of the alleles. And then Q is going to be the frequency of, again, the recessive allele. Down here we're talking about individuals in that diploid state, and here we're just looking at the frequency of the alleles. So, given all that information, I think we're ready to solve a problem. So, I'm going to skip this one. Okay, so here's a practice problem. If 9% of an African population is born with a severe form of sickle cell anemia, what percentage of the population will be more resistant to malaria because they are heterozygous for the sickle cell gene? So in this case, individuals could be, where's my pen? Individuals could be big S. In that case, they would be normal, and they would not have any sickled red blood cells or misshapen red blood cells because of a point mutation that causes the hemoglobin molecule not to form correctly. They can be big S, little s, and in this case, they would be carriers. And these individuals are heterozygous, and so they have some sickled cells, and it turns out in areas where there's malaria, they are resistant to this malaria organism because it replicates in the red blood cells, and it can't do so in these misshaped red blood cells. So these guys might get malaria, these guys are resistant to malaria, and then finally our homozygous recessives, and they would have the sickle cell disease, and they would die from sickle cell. So there's a selection for these individuals. So I want to know what percent of the population would have this resistance to malaria if 9% of the population is this here. So Q squared equals the homozygous recessive condition. So Q squared equals 0.09. So the frequency of Q squared is 0.09. So I want to know the individuals that are big S, little s. Well, that's going to be equal to 2pq. That's the frequency of the heterozygous individuals. All right, I know Q squared. I need to find Q. I need to find P. So to find Q squared, I will take the square root of both sides of my equation, and I get that Q is equal to 0.3. I know that P plus Q equals 1, so P has to equal 0.7. And now I just have to plug into my equation. 2 times 0.7 times 0.3. And so 21 times 2 is going to be equal to a frequency of 0.42, or 42%, because they asked me what the question asked me for a percentage, so I need to give that answer as a percentage, so it is 42% of the population. So that is a Hardy-Weinberg problem. The next slide just gives you a website where you can do some more practice problems if you would like to. All right, I hope this helps.